Thank you. Yeah, thank you, Stephen, for the nice introduction. Good afternoon, everyone. My name is Kuan-Hao. I'm a fourth-year CS student, and today I will be sharing a project I'm collaborating with Calico on titled Unifying ChIP-exo DNA Binding and RNA-Seq Coverage Predictions with a Multi-Species Fungal Language Model. With that, let's get started. For the purpose of this talk, when I mention sequence models, we'll think of them as a black box that map one sequence to another. These models are composed of primitive layers where each layer simply takes an input sequence, fitting to the model, and the model will return the sequence of the same shape, but this can be easily converted to higher dimensional features. And many sequence models have been developed to satisfy the interface, particularly in the context of deep learning. These include many classical deep learning models such as recurrent neural networks or RNNs, or convolutional neural networks or CNNs, as well as more model architectures such as transformers. And all of these kind of satisfy the same interface. They map a sequence to another. For example, you can use a normalization layer fitting to the sequence models of your choice and use a linear or nonlinear activation function as well as a residual connections. And you can fairly easily, by just adapting into your machine learning model fairly easily of any interest sequence model layers that you want. And for this talk, I'm focusing on the approach of CNNs and transformer-based models, which have been proven very successful in capturing long-range dependencies and interactions in DNAs. So this generic deep learning sequence models can be used to solve many different types of data with different types of sequence data. For example, like modalities such as text, to more general time series data such as audio, images, and videos. Furthermore, in the application to proteomics and genomics, it has been applied to protein sequences, and this is a screenshot from Meta's ESNfold, or a new startup called Evolutionary Scale, and also the DNA sequences, and this is from DeepMind's and Calico's collaboration Informer model. And DNA sequence is the main topic that I will focus on today. And if you put them on a spectrum from discrete to continuous, I'm going to draw very rough distinctions between different types of sequence model. Now, many model architectures focuses on data such as video, audios, or more generic time series data. And what's common to all of these is that there's an underlying notion of time from which the sort of data is sampled from. And in contrast to continuous data, what I'm focusing on today is roughly more discrete. And very roughly, I'll call them discrete sequence because input comes in the form of discrete tokens like DNA nucleotides. Okay, so when it comes to using standard supervised deep learning, there are many applications focusing on predicting directly from the DNA sequences, particularly in the context of gene regulation. So back in 2015, DeepSync and DeepMind were the earliest work that introduced deep learning based models for DNA sequences. Since then, the field has been expanded to include other exciting works, such as using high-C data to predict 3D structures of genomes or predicting chromatin profiles. And most of these deep learning models are built around CNNs, but now we're seeing a shift toward transformer-based models. And we're not just stopped there at predicting epigenetic marks. We also want to be very ambitiously to directly predict gene expression RNA-Seq coverages directly from the DNA sequences. And these include the state-of-the-art models such as Informer and Borzoi. So here's an animation illustrating how Informer and Borzoi works. First, you one-hot encode your input sequence, fit them into the model, and the model predicts a series of genomic tracks like ChIP-exo, RNA-Seq coverages. And to train this model, you need to curate a data set of DNA sequences paired with their corresponding coverage tracks. Both models follow the current paradigm of supervised learnings. So you train on the labeled DNA sequences to learn the patterns and test them in the separate test set. And on the right, I'm introducing an alternate approach. Instead of using the standard supervised learning, we can leverage self-supervised learning to uncover underlying structures within the DNA sequences to learn those conserved motifs. So here, during the pre-training step, we're training only seeing the DNA sequences without any labels during this training. And the model that got trained during this step is often referred to as language models or foundation models. And after we pre-train a language model, you can load the weight in and adapt it to specific tasks of interest. And for this example, it's the RNA-Seq track prediction. And this process, known as fine-tuning, involves further training to update the model weights to the specific task of interest, similar to what supervised learning does. Okay, so there has been a growing body of work applying language model to combine, particularly in protein sequences. Notable examples include GPT-2, ESM-Fold from Meta, and also ProGen2 from Salesforce and Profluent, which have demonstrated their power in protein folding, synthetic protein generation, and antibody design. And for DNA, I've highlighted some key language model papers here, for example, like DNA Bird, GPN, Nucleotide Transformer, Hyena DNA, Evo, and Caduceus, and more. Well, these works make significant progress, but in my opinion, the application of DNA language model is not yet as mature as or as extensively explored as protein language models. So what are we trying to build here? So in this study, we want to build a state-of-the-art gene expression model for yeast. And in the first part, I'll discuss some of our exploration on the DNA language model, and then our effort to fine-tune the language model and trying to answer the question if self-supervised learning with fine-tuning can do better than training from scratch. And we also want to understand the model, so model interpretability is also the main focus in this study. And then we can do a lot of applications, for example, try to predict the influence of the distal regulatory elements on the gene expression, and also evaluate the EQTL data. So why yeast? Because on human, the computation is very expensive. Here, I just do some simple math, and this is the amount of RNA-seq coverage data that Informer model need to see and Borzoi model need to see. So for Borzoi, there's 7 TB amount of data that the model need to see during training. And in terms of practical time, for Informer, it's trained on 64 cores of TPU, each with 16 gigabytes of memory, and it takes around 3 days. And for Borzoi, it used 2 800, each with 40 gigabytes of memory, and it takes around 25 days. So it's very expensive to do this training process on human. And furthermore, in human, we don't have a large-scale transcription factor perturbation data. And furthermore, yeast is a great model organism with all this benefit, for example, rapid growth and easy culturing, and we can simply do the RNA-seq to read out the gene expression at each time point. Furthermore, yeast has the conserved regulatory mechanisms, and last but not least, it's a great species to study aging. So that's why we choose yeast. Okay, and language model. What is language model? Here are some real-life applications, for example, like when you type, there's some next word predictions, and email suggested reply, or ChatGPT that everyone is using nowadays. So what language model, in the context of genomics, what language model does is you mask out some of the nucleotide in the DNA sequences, and the model tries to do this prediction based on the unmasked previous context and try to predict those masked nucleotide of its probability distribution of A, C, G, or T. So we're directly training a model on modernals, and if you think more carefully, actually, this training process is trying to learn the full joint distribution of the language. So here's a very rough definition of language model. It's learning the probability distribution over language sequences. Okay, I'll start talking about some of the fungal language model that we built. So here's a simplified cartoon representation of the yeast genome with 3 genes and repetitive regions highlighted. What we did is we chunked the sequences for each 16K with the step size 4K, and we can concatenate them into a tensor and do some data control, remove some of the repetitive sequences, and split them into training, validation, and testing. Okay, so after we create the data, the first question we ask is, why are we building a fungal language model instead of a yeast language model? The reason behind it is because yeast genome is very small. And also, there are thousands of fungal genomes out there with high quality. However, they don't have the supervised measurement, for example, like RNA-seq expression data. So what we're aiming to do here is we want to build a language model that pre-trained on all available genomes, and then we can do transfer learnings and transfer this knowledge to the yeast genome. So this is why we choose to build a fungal language model. And in order to build a high-quality language model, there are a bunch of questions we can ask. First is, to what evolutionary distance should we include into our LM? So actually we did a bunch of different dataset experiment so we can train on a single reference yeast and then ask the question that how about if we include more diverse strands of yeast, does it do better? So this is the second dataset that we have trained on 80 strands of yeast. And we also ask if it's possible that we further push more evolution distant genomes, for example, 165 Saccharomyces genomes in the older level, and finally push all the way to kingdom level, so include 1,000 fungus genomes to train this language model. I'll just tell you the conclusion. So the best performed model is this. So this is the final model that we go forward. And more question to ask is, for this trunking approach, we're adopting 16K, but is 16K good enough? And also, what's the quality of the annotation? And what's the ratio of protein coding genes? So here I just quickly go through and give you the answer. So in yeast, 16K window is a good window size. It captures on average around 9 genes per window. And in terms of annotation, we use buscode to evaluate the ortholog completeness, and it's around 95% completeness. So genome annotation is pretty good in fungi as well. And in terms of coding to non-coding ratios, actually, yeast is a very compact genome. So over 70% of the regions are coding compared to human, probably just 1% to 2% are protein coding regions. So this is also a very distinct difference between human and yeast. And then another important question to ask is homologous sequences. So overlooking homologous sequences between training and testing dataset is a common issue, especially for those ML researchers without biology background. So similar sequences across chromosomes are common and can cause data leakage, inflating the model performance. So what we did is, after we split into training, validation, and testing, we used Minimap to do the DNA level alignment, try to capture some homologous sequences between them. And here's just give you a taste of sequence similarities between testing and training in different chromosomes in different data sets. R64, 80 strains of yeast, and also 165 Saccharomyces genomes with the amount of sequences that were removed. I also just want to mention here that deciding how to strictly remove homologous sequences from testing remains an open question. So we can either perform the alignment at the DNA level or protein level, but removing similar sequences in some extent is crucial for fair evaluation and preventing data leakage. Okay, model architecture. So we actually tested a bunch of different models from smaller to bigger, with starting from simply just convolutional neural networks and to fancier model architectures, transformer-based UNet architecture. And I'll just tell you the answer. The model that we go forward with is the transformer-based UNet. So here's a cartoon illustrating how the model works. So first you input a 16K window into the model, and you need to reverse complement because we don't want the model to just learn the forward strand. The reverse strand as well is also important. And we're fitting to several convolution layers, scale down to 128 base pair resolution, feeding to the transformer blocks, and further scale it back up to 1 base pair resolution with a UNet connections. And the final output of the language model is going to be a matrix of 16K times 4 with prediction of each position if in the A, C, G, or T. So this is the language model. And here I'm showing the training and validation loss curves across page update. For this test set, we can clearly see the overfitting if we train on a single yeast and 80 strands of yeast. This is expected because genome size is very small. Even when we include more strands of yeast, the sequences are so similar, so it cannot generalize well. And the best performed model that we tested is the UNet small model trained on 165 Saccharomyces genomes, and this is the model that we go forward. Okay, so far I've only just shown a single matrix, mask language model loss, to summarize the model performance. But what did the model actually learn, and how did the motif prediction look like? So to predict and to understand what the model truly learned, we can analyze the conserved motif to inference the motif to see what the model predicts. And also we can visualize the attention maps to see what are the regions that the model focuses on in the self-attention layers. So to construct motif learned by the language model, we start by feeding the sequence in the held out test set into the model. We then mask out 50% of the sequences at a time and then the model to predict those masked regions. And then we mask another 50% and repeat this process iteratively until the PWN matrix was constructed. Once we have the PWN, we can calculate the conservation score. First, we add the pseudocount, do the row normalization, and entropy calculation, and also finally the conservation score is calculated. Okay, so let's take a look at some examples of the motif that the fungal language model constructed. So we randomly selected a few ribosomal proteins that focus on the promoter regions, 450 base pair upstream and 50 pair downstream. And in this example, you can clearly see that the language model learned the start codon. And upstream the start codon around 200 base pair, you can see the model learns the RAP1 TF motif binding site and also the FHL1 binding site. And these are all essential transcription factors that regulate the gene expression. And we can also see these patterns in other sequences that we investigated. So I was really excited when I first saw this motifs popped up. The model only saw shuffled trunk 16K DNA sequences from 165 sacromacetylus genomes during training. Yet, by using the self-supervised learning approach, it not only located the gene position, but also learned those conserved regulatory motif in the intergenic regions. So this is very exciting to me. And here's another example. In the upstream promoter regions of the SMT3 genes, our fungal language model is able to reconstruct CBF1, TYE7, and RAP1. These are all essential motif as well. Yes, once again, our fungal language model was able to identify these motifs through self-supervised learning approach itself. And here we can just simply more systematically find a motif in the genome. So we ran TF-MoDisco. It's an algorithm developed by Kondoji Group at Stanford. It's designed to de novo cluster PWMs into motif. And then we can compare this cluster motif with the East motif database like East Track and so on. And excitingly, we can see that the language model is able to capture quite a lot of interesting motifs. And this led us to another question that, can the fungal language model reconstruct motif in the genomes that it's never seen before? So we tested on 8 strands of yeast that never seen during training, and also 8 schizo sacromacetylus genomes that are in a different order level. And surprisingly, we can see that the fungal language model generalized pretty well and finds some fascinating motifs. And last, what exactly did the model learn at predicting these motifs? So here I'm visualizing the attention maps averaged across 8 heights from the last 2 transformal layers. And this example shows in the region of chromosome 12. What's fascinating in this fungal language model, it seems like the model grouped genes into distinct squares that I highlighted. And even more interesting, it focuses on a lot of intergenic regions, which are we assume that there are regulatory elements. So the model needs to check the attention maps when the sequence go through the language model. It has to focus on these regulatory elements in order to do the prediction. So this suggests intuitively, if you want to do, like, for example, predict RNA-seq coverage from the DNA sequences, having an understanding of the gene structure in the DNA is essential prior knowledge. And the attention maps here suggest that the fungal language model sort of learned the gene structures within DNA sequences. And this led to us to the question that, I'll quickly just show one last result I found really interesting. So, and then later we fine-tuned the language model and we did 8-fold cross-validation. And the solid curve are those models training from scratch, and the dotted curve are those models fine-tuned from the language model. And we can clearly see a substantial drop across 8-fold. So it suggests that language model helps the RNA-seq prediction. And there are more directions and results that we're exploring now. So thank you, everyone. I'd like to thank all the great collaborators and friends at Calico and also my advisors, Stephen and Ella. Lastly, I want to mention that we have a student-led study group on deep learning in genomics, and we meet every other Tuesday. So we just had a meeting yesterday, and the next one going to be come up on January 28th. So if you're interested, you can join the Slack channel and also come join us on January 28th. Thank you.